Okay, so I'm going to try to go over this as quickly as possible. I always say that, don't I? So we're going to hit so mainly what we're talking about here, we're talking about sensory systems. So we're going to hit some of the general properties of sensory systems, how signals are transmitted, that kind of thing. We'll talk about a concept called proprioception briefly, and then we'll discuss some of the somatic senses, which is body senses, which is touch primarily. And then the special senses, which are the ones we think of like eyesight and hearing and things like that. Okay, um, so to start with, there's a difference. We've always heard that there are five senses. We have five senses, and usually we think of sight, hearing, taste, smell, and touch. However, you can see that these lines are a little blurred. So we have way more than five senses. We sense things like temperature, which you could put in the category of touch. But we're also testing chemicals. We have chemoreceptors, which are testing, our oxygen levels and things like that. And these count as senses. So this idea that there are five senses is kind of a misnomer, because there really aren't. And instead of touch being involved in the special senses, we see that equilibrium is one of the senses, and that's certainly something that can't be categorized with the rest of these, and yet it's something that we're certainly sensing, and it's something that we're certainly aware of. So you can see that the special senses are these that are trying to get some kind of information from the external world. And then we have somatic senses. Somatic senses are just things that are involved with the body. Soma means body. Okay, so these are things that we're actually you can see touch, which we kind of understand what that is. Temperature, pain, itch, and then proprioception, and that's just awareness of body position. So that's kind of an important of body, and that's position. Okay, so that's kind of something that we have to get used to. We have to get used to using that word, proprioception. Okay, so like I said, we know everything. Chemoreceptors are testing chemicals. Mechanoreceptors, now when you think about what happens with hearing, um, we think, well, do we have a sound receptor? There isn't anything in our body that specifically measures sound. What it does is it detects vibration. Okay, so that means that there's a receptor that somehow can see when there is movement. Okay, so if you usually, this is actually the way it is, there are these little hair cells, and then when those are moved back or in forth, it'll actually cause an action potential to occur, and then that can be detected. Okay, and that can be interpreted by the brain. We have photoreceptors, photo meaning light, so that's involved with vision primarily, and then thermoreceptors, which are detecting temperature. Okay, so subconscious sensory is very significant. We wouldn't be able to function very well without it. We have certain things in our muscles called muscle spindles that detect stretch, and then Golgi tendon organs, which also detect stretch. One of them reacts when something is to get the position. So muscle spindles can tell you more about the position. Then Golgi tendon organs, uh, they can monitor when the stretching is too fast, too far, too fast, and then they'll react to that. Okay, so one is contained actually in the muscle spindles, and the Golgi tendon organs are found in the tendons, and these contribute to proprioception. So to distinguish and to figure out, I mean, if you were to look down right now, you know what position, say, your foot is in. Right now, you're aware of it, but your body is constantly sending feedback to the brain saying this is the position I'm in. Sometimes it fades, and you may sit for a while and it will go away, that sensation, but you can usually bring it back pretty quickly. Okay, all right, so and then we have this visceral stimuli. So these were part of somatic. So muscle length and tension, proprioception. That's part of somatic, understanding what's going on with your body, um, in terms of the physical parts of your muscles and that kind of thing, and your skeleton. Visceral stimuli, visceral refers to things like your internal organs and things like that. So you're constantly measuring your blood pressure. Your body knows what your blood pressure is right now and it's constantly adjusting it. Distension or stretching of the GI tract. So as things move through your gut, it's reacting and it's squeezing where it has to and it's relaxing where it doesn't have to, and you're not aware of that. That's subconscious. Blood glucose concentrations, your internal body temperature, your osmolarity of body fluids. We're constantly sensing that we have too much or not enough salt, sodium chloride, potassium, those kinds of things. We're constantly testing that and measuring that, and then we get thirsty if our osmolarity is too high, and we tend to dump fluids through our kidneys when our osmolarity is too low, when things are too dilute. Okay, lung inflation. We know how much that's taking place, and we can adjust that. The pH of the cerebral spinal fluid, the pH of our blood. We're constantly measuring hydrogen content. That's what pH is, and so we're constantly measuring that stuff. So these are most of these are chemoreceptors, and then you have your thermoreceptors here. Um, and then distension would be a mechanical receptor. Okay, so in general, if we look at this, we have to have some kind of stimulus for all of these that we've just discussed. We have to have some kind of stimulus, which means we have to have some kind of a receptor. Because the magic that's taking place here is we have things like light and sound and touch and chemicals, but that's not the way the body processes information. The body processes information through neuronal signals. Okay, so we have to turn all of that into some kind of a neuronal signal, some kind of an electrochemical signal. Okay, and so that's what receptors do. They get that information and then they turn it into this electrochemical signal. Um, and they do that usually through some kind of movement or something, chemical change that causes a change in the membrane potential. Now remember learning about the membrane potential, it's usually at about minus 70 millivolts. Well, when these receptors are activated, that will increase usually. And if we plot this out, so here we are and before something has touched our skin, and then in that particular area, the stimulus causes an increase. Now, if that increase goes over threshold, then we have an action potential. Okay? If that stimulus stays over threshold, then we can have repeated action potentials. Okay? And that tells us that that particular area is being stimulated. Now, this is the same with touch. This is the same with vision, hearing, all of those things. This is what ultimately is happening. And the more intense, the closer these action potentials are together. Okay? Now this information, as it gets to the brain, it will measure this intensity and it will say, okay, this is a very lightly intensive stimulus. Okay, so that's not going to register very much. That means this is barely hitting threshold, and so it's not firing very quickly. Or it could be very intense, and they could be firing one repeatedly right after the other, and then your brain would interpret that as being an intense stimulus. Okay, so how does this happen? We have, if we look at some of the neurons that are involved, we can see here that we have a few different ideas. I mean, there are just simply free nerve endings that are out there, and they have certain receptors on them or embedded into them that will react when there's a certain stimulus like fire or heat or something like that. They can react and then they can pass that action potential down the axon, and then that can be processed onto the brain, okay, or a reflex, and that information can go back and we can draw away to the muscles. Okay, we also have very specialized, this looks like a Pacinian corpuscle, that's what that's called, and that might sit just under the skin, and then when there's pressure put on that, if we were to push on that with like a metal rod or something like that, then that might cause some kind of a reaction here. So it's going to be mechanical in nature, and then that will send a signal. Now you can see that this is myelinated, okay? And we know that if it's myelinated, it's going to be fast, Okay? And generally, our sensory information is fast. Now, so some of these free nerve endings, it's not passing the signal very far, and so these could be actually unmyelinated, or they may be myelinated. Um, it all depends on their use and how quickly they need to process that information, and essentially how far it has to go. Um, Now here's another one. This is a hair cell. This is involved in things like hearing. So when you make something, it's usually attached to something called an organ of corti, and it causes vibrations in a membrane. And then these things will actually fold, move back and forth to detect movement of this membrane, that vibration. Or as you turn your head, this may be embedded in fluid. And as you turn your head, the fluid may move, and it may cause these things to wave back and forth, and that can tell you how you're moving your head, and that's what happens in the vestibular system. Okay, so I mentioned chemoreceptors, and this is what oxygen, pH, organic molecules like glucose, mechanoreceptors, I've mentioned that, pressure, stretch, uh, osmotic receptors can be mechanoreceptors, vibration, acceleration, sound. And so we're trying to develop a way that we can kind of understand this, because that's really the core, is that we have to take a mechanical signal or a chemical signal and turn it into an action potential. Okay, photoreceptors are specific to photons of light. That's very interesting. All you have to do is somehow have wavelengths of light, which light carries energy, to cause some kind of a change in the cell, and that's where we have our rods and our cones. Okay, so rods and cones, and then that will cause the light coming down, and hitting that will actually cause an action potential to take place. It's always converted into that. Thermoreceptors are sensitive to different amounts of heat, okay, or cold. All right, so the stimulus energy, whatever energy this was, whether it's vibration or light or whatever, is converted into information and processed by the central nervous system. So we have ion channels, second messengers. Remember that was G proteins. Um, and those things will either an ion channel or open, or somehow this stimulus energy here will cause some kind of change in a receptor or a channel or some other kind of cellular process. And if it's and we have an adequate stimulus, which means that it would be specific for that particular modality, or whether it's temperature or whether it's light, the form of energy to which it is most responsive. Okay, and then we mentioned threshold. Okay, threshold is that minimum stimulus. You can, if you touch your finger with a feather and barely touch it, you may not feel anything at all. But then as you push harder and harder and harder, then it starts to activate those mechanical receptors, and you have sensation. And then that, as I said, changes the receptor potential, and that signal then is moved on. Okay, so once we have that signal. This is showing some of the special senses. Gustation, which is taste. That always is processed by the brain. Usually this information, for special senses anyway, comes in through cranial nerves. I don't know how to spell cranial. Cranial nerves. Okay. And that's primarily special senses, not always, because we have like the vagus nerve that is that takes information about digestion and sends that to the brain. But primarily we have like the vestibulocochlear nerve, which is bringing information from hearing and the vestibular system, and we have like the optic nerve, which is bringing information here. And so we have the eye, and if you let's look at something. Here's the olfactory nerve here. Let's look at something with all of these, and this may not be too significant, but I'll tell you why it is significant. Is that each of these will go through an area. If you look at these, they're all sort of collecting on this little yellow thing right here. and that's the thalamus. And if we look at this, our taste goes through the thalamus. Somatic senses, touch, that kind of thing, goes through the thalamus. Vestibular, so our equilibrium, through the thalamus. Sound, through the thalamus. Sight, everything, except smell. Smell is actually processed here at the olfactory cortex. Why is that significant? Because the olfactory cortex is hooked directly with emotion. And so a lot of times when we smell something, we may actually have an emotional response before this information is then passed on to the rest of the brain and we're actually conscious of what it is. And that message may go on through the thalamus as well. But this is where the identity of it is processed. However, this is linked directly with emotion, and so this is more common in animals where they smell something and that information is then uh, evokes an emotional response nearly immediately. And humans have some of this left too. Okay. All right. So the properties of the stimulus, which is this modality, which is some kind of information that it's picked up on, whether it's temperature or smell or something like that. Um, so this happens wherever the sensory neurons are activated. So we don't hear sound through our hands. We feel touch, and same with smell and vision. Vision is in our eyes. But these neurons, as we just saw, always terminate in the brain, and that's where it's processed. So each receptor is most sensitive to a particular modality of stimulus. So this means that we have a lot of different sensors, and each one of those is fine-tuned to pick up on that particular modality or that particular sense. Okay, so this is the other thing that I was mentioning earlier, is that stimulus, intensity, and duration. So that's kind of a big deal, and this is the thing that you have to understand, because when we're talking about converting something to a chemical or a neurochemical or a electrochemical signal, um, it's hard to say the intensity. There has to be a way to have the intensity, because remember, action potentials are all or nothing. So that means we can't have a stronger action potential. So how do we handle that? The simplest way to understand this is we have an increase in frequency. Okay? So I hope that makes sense. If we have a very small signal that barely reaches threshold, and this is what I was drawing earlier, it may be fairly slow coming across. It may depolarize or may repolarize a little bit before it's activated again. And that means that the signal isn't really big. Okay, so you're not being poked too hard if it's going very, very slowly. However, if it's very fast, then our brains will say, wow, that's really coming in at a pretty good clip. And then you will consciously, unless it's one of these subconscious internal types of stimuli, but you will, let's say it's something that you are aware of, you will consciously say, wow, that's a very intense signal. And so if it's coming through very slow and this neuron is being activated, so if we're activating this neuron, then a little bit later activating again, of course we're talking, this is seconds, so this is, once every second, and this is many, many times per second down here, the fast one. But if we're activating this, then, we activate it a little bit, we'll release a little bit of neurotransmitter. But if we're continuously keeping this neuron here active, then we're just going to keep releasing more and more neurotransmitter, and that's going to keep the next neuron very, very active. And that can be then processed in the brain as a high activity of these neuronal signals. Okay, so let's kind of go through. We're going to first talk about some of the somatic senses. Um, Touch, and as we mentioned, proprioception, which is essentially body position. Um, Temperature, and then nociception. So this term here, nociception. And that really, mainly, I'll tell you, when we talk about this, we're usually talking about pain. Okay? Now, itch is a form of pain. It's pain that's not intense enough to be pain, but it's intense enough to activate this other neuron called the C neuron or C peptide. And that's what causes itch. So pain and itch are actually fairly closely related. We're not going to get into that too much in this class, but they're fairly closely related. They share some of the same neurons. Okay, so if we see this, we see here fine touch, proprioception, vibration, nociception, temperature, coarse touch. Okay, so we have a couple of different pathways that these can take. We have information from fine touch and vibration that goes directly up, and we're not going to get into what these different pathways are, but we can see that there are a couple, and that either way, our goal is to go through this thalamus, and then that's processed in a certain part of the brain called the sensory cortex. Now let's think about what that means. So what that means is that when you, let's say, if you look at how the brain is mapped, there's a certain area, and it runs right along. So if you have a top-down view of the brain, okay, there's this area called the central sulcus, and it sits right here. Okay, all the way across the top of your brain, and it just goes all the way down. So this is like a cut through the middle of your brain. That's what you're seeing here. Okay, so if we were to, this is the top-down view. If we were to cut it just like this and then turn it, this is what you would see, and you would see that it is all along this area here. And it isn't just um, random the way that it's set up. We can see that it's actually very specific. So this part of your brain, in this uh, motor cortex, is specifically dedicated to the feet. Okay, and then you have a huge area dedicated to the hands, and we have a huge area dedicated to the face, particularly the lips. So these are obviously very important for sensation. A significant part of the brain is dedicated just to the hands and the face. I mean, you could say it's almost half. It's at least over a third is just for the hands and the face. And then we have very little for the back and the torso area. Um, and then we have some for the legs, but not nearly as much. Okay, and then the genitals are over here. Kind of a funny story. There was some scientist at one time saying that the reason people have foot fetishes is because the genitals are so closely linked to the feet in the brain. Now I don't know. That sounds almost like I don't know. That sounds like one of those things that sounds like science, but I'm not sure if that's true or not. Okay, so we're talking about somatosensory. So we're talking about touch, and we're not going to get into all of the details and specifying all of these different types, but we have several different types. I'm not going to test you on the different types and what they do. I guess sensory nerves, they all come down to the sensory nerve. Um, but we have a lot of different types of receptors. So Pacinian corpuscle, Rossini corpuscle, Merkel receptors, Meissner corpuscles, and these are all scattered through the skin. And some of them can detect fine differences, like if you poke there versus here, you can get actually a distinction between these. Now we're better at that on our hands of distinguishing between 2 points. It's point discrimination is what it's called. Between 2 points on our legs, like on your thigh or something like that, you're not going to have that kind of sensitivity. You may poke here and feel the same thing. You may not be able to tell. You may have to go like an inch away before you can tell you're poking in a different place. Now that's not true on our hands. So these vary a lot. Okay, on our skin, the distance between them, how much branching or convergence takes place, because we're going in this direction. They converge onto one, and if all of these converge onto one, then they're all going to be detected. If you poke at this one, it's the same as if you're poking at this one. You may have a more intense signal, but it's all going to be registered the same. And then we have these that are deep down in, uh, that are going to measure pressure, as opposed to light touch, which you'll have these nerve endings up here to measure light touch. Okay, temperature receptors. These are actually kind of interesting. They're always free nerve endings, so that means they don't have one of those, like the Pacinian corpuscles have these head groups on them. Um, so thermoreceptors tend to be, or temperature receptors tend to be free nerve endings. And so we can feel hot and we can feel cold, and all they are is they're just receptors that are activated at different temperatures. I mean, that seems logical. Um, and we see this, and we can test these because your cold receptors, okay, so these are just receptors with regular um, regular types of receptors on them, mechanical receptors or chemical receptors. But these are activated by, these happen to be activated by menthol. So cold receptors are activated by menthol, and that's why when you have something with mint, it tends to feel cool. Okay. And it will detect anything lower than body temperature. So when they're activated the way they're supposed to be, they detect different temperatures. However, we do have them that can be activated by this menthol. Hot receptors, or warm receptors, these are activated above body temperature. So anything above your normal body temperature, which is about 37 degrees Celsius, through about 45, that feels warm. It's not particularly painful, it just feels warm to you. However, above that, it also activates pain receptors. Okay, so these thermoreceptors, or temperature receptors, if it's above 45 degrees, and of course that might be a little different for everybody, but on average about 45 degrees, um, then you will actually feel pain. That temperature will start to feel like it's a painful response and something you want to get away from. Now, the other interesting about this is it's activated. These are just, as it turns out, capsaicin will open these receptors, will activate these receptors chemically, as opposed to just being a temperature change. And capsaicin, I don't know if you're aware of this, but that's what's found in things like peppers. I'm trying to draw pepper here. Um, that's the worst horrible pepper that has ever been drawn. So uh, that's a pepper. Okay, and peppers have capsaicin. Okay, and so that's why peppers feel hot to us, because they activate our heat receptors directly. Okay, nociception. Uh, like I said, pain, primarily pain and itch, respond to noxious stimuli. Okay, so the whole point of nociception is to avoid harm. Okay, so when the tissue starts to be damaged, or it's activated in such a way where it could be damaged, uh, nociceptors will be activated, and your body will interpret that as pain. Okay, and then you will have some kind of a reflex, or you will have some kind of a desire to move away from it. People who, it's very rare, but there are people that have an inactive nociceptor pathway and they don't feel pain. And that's actually very, very bad. They usually end up dying at an early age. They'll break their leg and they'll just be sort of running along with their leg dangling, because they don't feel the pain from that, and that's actually not a good thing. So it's good that we feel pain. It keeps us safe. Okay. All right, and now let's start talking about some of the special senses, and we're going to go through these pretty quickly because we just need to have an idea of what they are. The olfactory system. Now, the olfactory system tends to operate on chemical. Here, I'll just write that out. Chemical receptors. Okay. So different chemicals in our environment. We smell something, everything that, if you smell, I was going to try to draw an orange, maybe that's safe. So if we have an orange or an apple, or whatever you want to make that, it will release certain odorants, certain odorant molecules. And those little molecules can go in through our nostrils, and they will bind to a receptor. And we have a whole bunch of different receptors for different odorants. Now, that doesn't mean that for every smell there's a different receptor. Usually it's a combination, and these will come together, and we can have, say, something else. Maybe this is giving off several different odorants, and the combination in which they activate the nociceptors, or I'm sorry, the olfactory receptors, is will give a signature, and we'll say, okay, that's an orange. We know that. Um, and so that's processed here. It's processed in the olfactory cortex, which is very, very close. And then, like as I said, this is the only one that doesn't go through the thalamus, and it may be processed, and you may then become aware of what it is. Okay. Um, And so we can see this a little bit more closely here. We can see here, down here, are a bunch of cilia, and these all have, I don't know if you can see this very well, but they all have receptors on them, okay? And they're just covered with receptors, and each one of these will then go through and it will process, and this one will be specific to a specific smell um, or odor, and then together, whatever's active, let's say this one might be activated here, and this one is activated here. These will go in, and then they will move through the olfactory nerve to the cortex, and we'll say, okay, I know that combination. That means we smell whatever that is, an orange. Okay, taste is pretty much the same way. In fact, taste will work alongside olfaction to help your brain identify certain flavors. We know that if we hold our nose, taste just isn't the same, and so there is a combination and some sharing of those chemicals. Uh, but we essentially have five basic sensations when it comes to gustation. Sweet, so that's let's go through these and let's think about what we're getting out of this. Sweet means sugar, okay? Sugar is energy, okay? Your body needs energy, and the easiest form for it to process is sugar, okay? Yes, there is also energy in fat, and there's energy in proteins, but we don't really use protein energy unless you're starving. But its favorite form is sugar, and that's why sugar usually tastes very good to us. In the wild, it's much more difficult to find something sweet. In the grocery store, it's pretty easy, which is a big problem with diabetes and obesity. Okay, and then we have sour. Sour usually means some kind of acid or pH. So if you have hydrogen, that tends to be sour. So we have so that's actually useful. Salty. Well, sodium. Okay, we know we need sodium. Think about how action potentials work by sodium and potassium. Okay, so potassium and sodium will actually activate salt receptors directly, very directly. I mean, when you think about what happens with an action potential and that sodium rushes in to depolarize it, all you have to do is have a little sodium channel sitting on your tongue, and when you get sodium, it directly activates that, and you have an action potential, and it lets your brain know that there's sodium in your mouth, okay? So the other one, or another one, is bitter. And when we think of bitter, we think of poison. Okay, so that's skull and crossbones right there. I think I can draw just fine as long as I tell you what I'm drawing. So this is poison, and this is danger. Okay, so that's the idea. Now we know that we all the time eat things that are bitter. We've tested them, and a lot of times there are nutrients in things that are bitter. But our taste receptors kind of want to err on the side of caution, and so they want to deter you from having something that's very, very bitter. And so even though we can override that, if you're left to the wild, if you taste anything that's bitter, the chances are that it could be toxic. Okay, and so we've got sugar, um, pH changes, we have salts, all of these things that are necessary, and then we have this other flavor called umami. Umami, you may have heard of MSG, or monosodium glutamate, which monosodium glutamate is hardly anything except glutamate and a sodium, okay, hooked together. Okay, mono, sodium, mono meaning one, sodium glutamate. Now, if you don't know what glutamate is an amino acid. So it could be, and the flavor that we see is a lot of times usually referred to as savory. It's usually used as a flavor enhancer or something like that. We hear a lot of bad things about it, and I'm not going to challenge that, but I do know that it's simply glutamate and sodium. And so this is our desire to bring in amino acids. Okay, so we need amino acids to function, and so we have a receptor for amino acids essentially. Okay, so each of these is sensitive to only one taste, and sometimes if we have a deficiency in something. A lot of times I myself have desired something. I need something salty. Um, or even sour. Sometimes I do that. I get in that kind of a mentality too. And usually that's because your body is sending out a signal saying, hey, your salt levels are getting low. So it's going to ask you to go find some salty stuff. Okay, so our taste buds, we always hear about taste buds, and see this guy. Let's trace one of these. Here's sweet. It can be activated. This will go down, and then it will actually activate a neuron. So the one that they're using as the example is sour. Goes down, activates the neuron, releases a neurotransmitter, and then sends it off to the brain. Okay, and move that back down. Sends it off to the brain, and then that can be processed. All right. So let's talk about the ear and hearing. Now, the hearing, we're talking about sound vibrations. When we think about sound vibrations, we usually think about waves, but it's really not waves. It's like um, dense sound and then thin sound and then dense sound and thin sound, dense, thin. And so it comes in, if we look at that, I started too far away. Da, 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 da. Dense sound, dense sound. What that's going to do as it hits, it's going to hit hard. This is the tympanic membrane. This is your eardrum. It's going to hit it hard, and then it's going to hit it softer, and then harder, and then softer. And that's what the wave really is as it comes in. And so it's going to cause this tympanic membrane to vibrate, which is going to cause these guys to increase that signal. Okay, so these are the bones of your inner ear, um, and then that's going to activate a membrane that rests right inside the cochlea. Okay, so there's a membrane in there, and if it's a fast vibration, it'll activate it here. If it's a slow vibration, oops, it'll activate it here. Okay, so then we can tell the difference between whether it's a high frequency sound or if it's a low frequency sound. A high frequency sound will activate here. Then once we have that, we can actually distinguish between different frequencies by where it vibrates, this little membrane that sits inside the cochlea. So then all we have to do is put a little receptor here. And if we say this receptor is activated, so we can have the auditory nerve that's moving out. If we say this receptor is being activated, then we know that it's a high frequency sound. It's this one, so we can put another one here. And of course, it's not going to just run through like I'm about to draw. And we can say if this one is activated, it's a low frequency sound. Okay? And so what we've done is we've just taken sound, turned it into something mechanical, and then we can have the vibration of this membrane, and all we have to do is have a mechanical receptor right here that's detecting that vibration and sends a signal to the brain. Okay? And then we have our sound that is processed. Okay? So if we look at this a little bit more closely, this is what we see. We see this is the sound. This is what I was trying to draw with the dense versus thin or less dense air. When you clap, you're going to push all of the air just as your hands hit together, all of the air at that one time, and then it's going to be less air. So we're causing dense areas of air, and then less dense areas. So that moves, shakes the tympanic membrane. It's actually quite simple. As the air moves in, it's shaking this, which is causing these guys to vibrate, which then causes, and this is kind of something I left out, the fluid to vibrate inside here. Okay, so there's fluid inside here, and this fluid will vibrate at whatever frequency this is vibrating, which is vibrating at whatever frequency this is vibrating. Okay, and so we have this amplification here, and then it vibrates this fluid, and so this is this membrane here. Okay, this membrane that sits around here. And it's called the basilar membrane. It's not labeled on here, and it's not something I'm going to ask you. But this membrane will then, let's see, this just calls it a flexible membrane. Fluid waves push on the flexible membranes of the cochlear duct. Okay, so right here, it's pushing on it at a certain frequency. This is whatever frequency, and then you just have receptors with little hair cells that get vibrated, and it turns that into a signal. Okay. Remember, that's the key, the neuronal signal. Okay? All right. So how do we tell the difference between left and right? Well, it's pretty easy. You have neurons that are processing the signals in from there, neurons that, here, let's draw it here where we can see it, neurons that are processing the signals in from there, neurons that are processing the signals in from there. So if it gets here first, it may end up there, and this one, let's see, if we draw these over a little bit, let me start that over. So if this one's coming in and it gets to that one first, it's moving as fast as it can. It gets all the way to here. This one comes in, but it took it a little longer to get there because it had to travel all this way first. So it didn't get there quite in time, and it goes to there. And then when that is processed, we can say, oh, it's on this side. Wait, that's the other, it's the other way around. So it would be saying that it took longer to get here, so it would indicate that the sound is actually coming from this direction because it moved this way faster, so your brain will switch that around. It's not exactly how it happens, but you get the idea. It will land that signal. These will converge on different areas based on where the sound comes from. If it's very quick to go in here, it may make it all the way over here before, if the sound is coming from this direction, before it travels all the way around, moves through here, and it may only get to this point here. So wherever they converge, that's where it says, oh, it was all the way on this side, so that means the sound is coming from that way. And we also have the shape of the head that shadows the sound and makes a difference as well. Okay, so vestibular. Okay, equilibrium. Now, this is similar. See, here are these hair cells. This is the same kind of thing that we were talking about with sound, that these guys can move, only it was vibration in the cochlea. It was vibration that was causing these to move in certain areas based on the frequency. Here, you have these tubes that are essentially in three planes. One would be like if you're moving your head shoulder to shoulder. Another one would be, this is going to be hard to draw, but as though you were moving your head, shaking your head, yes. Okay. And the other one would be as if you were shaking your head, no. Okay, so we have three different planes, and when you turn your head, it causes these guys to shift. Okay, so there's fluid in here, and we know that this is bone. That's going to move with your head. Well, the fluid isn't. The fluid will actually, just like if you have a bowl in front of you full of some kind of a liquid, if you turn the bowl, the fluid doesn't move right away. It moves a little bit, and that's what this detects. It detects that difference. It says, well, the bone just moved a whole lot, okay, around it, but the fluid resisted, and it caused this thing to go whoosh in this direction when that bone went this way. Okay, so when your head went this way, that fluid didn't move right away, and it pushed these hair cells that way, and magically that is a mechanical receptor. It detected that movement, and it sends it through a signal. Now you have, again, a neuronal signal. Okay, and that then can be processed by the vestibular part of your brain. Okay, now eyes and vision. Eyes are actually a little bit fun, a little more complicated. But light enters the eye, it focuses on this area in the back called the retina. Okay, and the lens is this thing in front that helps focus it, and we have muscles attached to the lens where we can make it a little more rounded, or we can make it a little less rounded depending on how close something is. So that's how we pull things into focus. We have photoreceptors that are kind of amazing, that will actually take light signal and again turn them into an electrical signal or electrochemical signal. Okay, because of course when we talk about electrical, we're talking about the movement of ions across. Okay, so remember that when we have an action potential, it's chemical because it's an ion, but it's also electrical. Okay, so these neuronal pathways process these signals into visual images. So that just has to move to the brain. Okay, so we see here. So we have the signal. It's processed back here, and this is the retina that sits around here. Okay, light comes in, we have peripheral vision, we have very direct vision that focuses right here on an area called the fovea, which is that focus, and then all of that information will faithfully, so we have neurons that go here, neurons that go here, and they all move back here, and they go to the occipital lobe, and they maintain where they came from. So if we see, you know, a stick man here, then that information, the stick man will be turned upside down, okay? That information will be put on the back of the eye, on the retina, and then that information, the leg, will be then processed back here, okay? It does cross over. So things on your left visual field will actually be processed on the right of your occipital lobe. And so there, yeah, everything's pretty much upside down and backwards as it's processed, but we're able to, but the brain's able to interpret that, and everything comes out okay. So if we look at this a little more specifically, this is where the light goes in, and this is the retina back here. Now, I mentioned the fovea, and this is the point here where you have the best vision. So this is where when you try to focus, you're trying to focus on the fovea. When you look at something very specifically, the most light and the most information is going directly to this fovea. Yes, you are processing things out here as well, but that's your peripheral vision. Okay, if you want the best acuity, you focus on that area right there. Here's the optic nerve. Now you can't see anything through the optic nerve. So if we look, let's see, up here and say this is where we're actually trying to focus. Wow, we can't see that at all, can we? Say this is where we're trying to focus, is here. This is the fovea. Well, this is where everything's leaving the eye, so we have no vision here whatsoever. And that's usually off to the side a little bit. This picture doesn't exactly show it that way. But this is usually off to the side of our vision a little bit, and you can find it if you put a couple of dots on a piece of paper and then move them closer to your face. You can see where one of the dots will disappear if you stare at one, and it's usually in our side vision. So that's usually how we recognize it. So if we're viewing something here, if you're looking at something, so if this is what you're looking at, and there are a couple of dots, and you stare at this one, this dot. So if we have a piece of paper, we put two dots on it, and you're staring with, let's say, your left eye, and you move this thing closer and closer and closer. Eventually, this one will disappear. Okay. Because that's where your optic nerve is going in, and you can't have any vision here. There's just simply no area for the retina to be, for the rods and cones that we'll talk about to be. Okay, so that's your blind spot, and then this fovea is where everything is focused. Now, we don't have to go through too much of this. I'll mainly just talk about the rods and the cones. Uh, but this information, you've got innervation, which is why diabetes can be so harmful, because you have very, very thin blood vessels that have to feed all of these things. Okay, we always need a blood supply everywhere, including in this area of the eye. So we have very thin capillaries, and unfortunately, high blood sugar can damage these capillaries and wipe them out, and that's what can cause blindness and diabetes. Or vision problems in general. And so the light has to, and the reason they have to be so thin is because the light has to penetrate all of these layers of cells to get to these guys. These are the ones that actually detect the light, okay? So we have the rods, which are shaped more like rods. So this is a rod right here. And then we have the cones that someone decided were shaped a little more like cones, okay? And that's what we see right here. The cones are for color, and the rods are for monochromatic. A lot of people just say black and white, but it doesn't detect color. Okay. Now, one of the things that's special about the fovea is that these cells, these nerve cells, these neurons, are kind of angled away, and so you've got this area cleared out, this dent that forms on the retina that lets as much light go through as possible. Okay, so remember what I said about rods and cones? Let's look at this. If here are the rods and cones in this layer right here, these are bipolar cells, and these are ganglion cells. We're not too worried about those. That's how the communication takes place to go to the optic nerve. But light is coming in from this direction. Don't ask me why the rods and cones are on the back instead of up here where they would receive more light in the first place, because I don't know. I don't know that anybody knows. But this is the way it is. So light comes in. So we bend these things around to get as much light as possible. But look at what this is. Remember, the yellow ones in this case are rods. The purple ones are cones. What do you see at the fovea that's special? You see a whole lot of cones. Okay. Now, cones will give us the most color vision. They give us the color vision, and they give us the best visual acuity. So you can pick things out the best in this area. And also, that's where the most light is centered. And that's important because cones need a whole lot of light. They need more light to activate them. They're very specific in their wavelength, so they're very picky. And so the light has to activate them, and it has to be within a specific wavelength. Okay? And so that makes them a little less sensitive to just overall light in general. Rods, they don't care. They can activate in whatever light. But this is a lot of times why when we're trying to look at the stars or something like that's very dim, we have to look away a little bit. We have to give these rods a chance to really process that information and get that information to them. Okay, photoreceptors. So that's what we call these, the rods and the cones. Rods function well in low light. This is why you cannot see color very well in low light, because you are mainly activating your rods. Okay? Cones, color. It's very handy because they both start with C. Responsible for high acuity. Okay, so that's really being able to focus on something and get a lot of detail from it. Okay, and then again, we have the same situation. Visual pigments convert light energy into a change in membrane potential. We're taking light energy, and so light energy coming in, and we're turning that into, so we have our receptors here, and there are receptors that respond to the light energy. The light activates them, and then we have an action potential. Okay. I'll say something else that's kind of interesting. Rods and cones contain um, a pigment that's basically made from vitamin A, and that's how they work. That's why we say that you have to have vitamin A for, it's called retinol, and so you have to have this vitamin A for good vision. Okay, because this molecule right here, it's active, that's activated by light, is actually retinol. Okay, and retinol is a form of vitamin A. And so if you don't have very much retinol, then you're not going to be as sensitive to light, and it's going to be harder to have an action potential. Okay. I have color blindness mentioned here, but color blindness just means that you usually have 3 different pigments. So you have the red, the green, and the blue. And in color blindness, maybe one of these is not functioning properly, and so you're not able to detect the colors the way that you would like. And so this is how it is. Here are your blue cones, and they detect color in the blue range. Okay, okay, they're not going to detect color up here in the orange and the red areas. And then you have the green, which tends to focus information in the green areas. Okay, so under this part, okay, so it gets this, and primarily around this area. Your brain will interpret those wavelengths as green, and then the red will primarily get this area. Now I know that this is highlighted in this zone, and you can't detect anything outside of this. Okay, so there are certain birds that will move. Actually, they tend to move off into the ultraviolet. So there are certain organisms that actually have photopigments, what we call these, that will actually go off into the ultraviolet, and they can actually detect ultraviolet colors. There's nothing wrong with that. It's all a big long spectrum. It goes all the way out to radio waves, or radio waves X-rays in this direction. Um, but we're limited to this range right here. Okay, so this is as far as we can detect this way, and this is as far as we can detect this way because of the rods and cones that we have. The rods, as you can see, kind of hit almost every part of the spectrum. They're very sensitive to light. Okay. And the brain just simply takes the combination of all of these and it interprets it. I say simply, but it's actually very, very complicated. So complicated that we don't entirely understand how it does it. But that's what it does. It takes a little bit of blue color and it says, okay, this much blue color is activated and this much green is activated. So we're going to call it whatever this color is here. Okay. All right. Sorry, I can't tell you more about that, but simply science doesn't entirely know. It knows more than I just said, but not completely. Okay, so this is what I said here. We have our visual field. Our visual field means that when we detect something here, it hits on this side of the eye. So I'm going to make this a little bit neater. So when we view something here and we're not looking directly at it, it's detected in our peripheral vision and it will then move, and this is the optic chiasm where it crosses over, and then it moves down here and it's processed on the opposite side. Okay, so we have that. Now if we see something in our peripheral vision this way, moves here, and then it will actually stay on this side. So that means each eye, if you cut the optic chiasm, you're still going to be able to see all the way around. Now I'm not going to test you on this, but I do teach classes where I do, um, just not this one. But you can see that we can take all of this information, and then the brain has this information and it's able to process it. Again, I could say, and it simply turns it into binocular vision, but that's kind of a misnomer because it's far beyond simple. Again, it's something we don't really understand. We know that it can take just a slightly different perspective. So if you're looking at something here and here, it can take the slightly different perspective and then it can process that. We call that an association area. So it'll get that information faithfully here and then it will say, hey, you know what? It looks a little different to me and to me, and it'll take that information together and it'll say, okay, and I'm going to interpret that back to the rest of the cortex, and I'm going to tell your brain that this is something that is this far away or has this kind of a texture or surface. Okay. And so we need that. We need both eyes to be able to do that. If you only have one eye, you're not going to have that coordination between both of these perspectives, and so you're just going to see the basic surface, the basic information, which is better than seeing nothing at all. Okay. All right. Okay. Well, that's it. That didn't take nearly as much time as I thought, or as afraid it would. So, but that's it. That more than covers everything I think that you're going to need to know for this particular chapter.